than in cultured GABAergic neurons. So now again, I have to do this. So this is another study that we did together with Leif Hertz. Namely, we looked at production of carbon dioxide from labeled glutamate and glutamine in cerebellar granule cells and in cerebral cortical neurons and in astrocytes. And you see that in the neurons, glutamate is metabolized actually quite rapidly, while in the astrocytes, glutamine is metabolized better than glutamate. So that's another important difference between astrocytes on one hand and different types of neurons on the other hand. Another thing that we did with Leif Hertz and a PhD student of his, Albert Yu, who is now residing in Beijing in China, we looked at the metabolism of C14 labeled glutamate in astrocytes. And we were curious to see whether this was metabolized primarily via glutamate dehydrogenase or via aspartate aminotransferase. And one way to look at this is to use the carbonyl trapping agent amino-oxyacetic acid. And as you can see here, the glutamate metabolism is inhibited by amino-oxyacetic acid, and so there's no aspartate formation. And strangely enough, the carbon dioxide formation was not inhibited by amino-oxyacetic acid. So you could say, well, maybe it's because it doesn't work. But the fact that aspartate is inhibited, aspartate aminotransferase is inhibited, is an indication that in the astrocytes, the oxidative metabolism of glutamate is primarily taking place via the action of glutamate dehydrogenase and not via a transamination reaction. And now, oops, what's going on here? So another thing that I stumbled upon just by chance, I was looking at the activity of different enzymes associated with tricarboxylic acid. And one of the enzymes is pyruvate carboxylase. And at that time, nobody knew anything about where this enzyme was expressed in the brain. So we looked at the activity of this enzyme in mouse brain, adult and newborn, and in astrocytes, and in the two types of neurons that we have in culture. And to our great surprise at the time, we could not measure activity of pyruvate carboxylase in the neurons. 1 year later, there was an immunohistochemical study published in Brain Research and showing the same result, namely that pyruvate carboxylase is an astrocytic enzyme. And that's a very important observation, and it was totally surprising at the time. And I'll come back to why this is important when I go into the metabolic studies in a little while. And it was not something that I had planned, I just did it. You never know. In the late 80s, I had a visitor from Greece, Georgios Palaiologos, and Georgios was interested in looking at whether or not aspartate aminotransferase might be involved in the biosynthesis of transmitter glutamate. So he did some very important experiments, Giorgio. And we could show, or Giorgio could show, that if you look at glutamate release from glutamatergic neurons, some of this release is actually transmitter related, some of it is just spontaneous release. And the the transmitter release can be monitored replacing calcium by cobalt. Cobalt is a calcium antagonist. So in the presence of cobalt, the transmitter release is inhibited. And so you see here that this is the case. When we look at this here, there's a decrease in glutamate release when we add cobalt, and this is not so clearly shown by aspartate. And more importantly, we could, more importantly, we could show that two compounds, namely again amino-oxalic acid and another one phenylsuccinate, two substances that will inhibit the malate aspartate shuttle, inhibiting the transaminase and the ketoglutarate, or the keto acid transferase. And you see here that the release of glutamate and aspartate was almost completely blocked. The transmitter release was completely blocked by these two inhibitors of the aspartate aminotransferase and the keto decarboxylase carrier. So that led us to suggest the following model for synthesis of transmitter glutamate. And as you can see here, it involves the malate aspartate shuttle and malate aspartate and the ketoglutarate, keto acid transferase. And the model is that you have, since aspartate aminotransferase is expressed both in the cytosol and in the mitochondria, that is capable of transaminating alpha-ketoglutarate to produce glutamate in the cytosol and to use glutamate in the mitochondria. And the glutamate comes from the degradation of glutamine to glutamate with the enzyme phosphate-activated glutaminase, which is present in the intramembrane space in the mitochondria. So this was a model that we published early on, and there was a recent review by Jerry Thienel and Doug Rossman in Journal Neurochemistry, just published a few months ago, where they looked at this in detail, and they have confirmed that this is indeed the way transmitter glutamate is synthesized. So we were pretty early on that time. Let's see. Yeah, another interesting thing that Leif Hertz and I did while sitting, having nothing better to do, we came up with this scheme that we have called a non-existing brain cell. All these reactions are present in the brain, but there is no cell that has the whole thing. You have to have collaboration between the cells to do this. One example again is the pyruvate carboxylation, which is up here. It's only at astrocytes, and you have the GABA metabolite, the GABA synthesizing enzyme, glutamate decarboxylase, which is here only in GABAergic neurons, and glutamine synthetase, another important enzyme in the brain, is only present in astrocytes, which was shown elegantly by Mike Nornberg and co-workers in the late 70s. And this, of course, is the tricarboxylic acid cycle, which is present in all cells. So the brain is complex, as you all know. And again, oops, I don't like this. It's doing this to me all the time. What's going on here? Here we go. Try again. Here we go. So this is the metabolic fluxes of glutamate and glutamine in the astrocytes. As you can see, astrocytes here, cerebellar granule cells and cerebral cortical neurons. And for those of you who may not have remember this, the cerebral cortical neurons are the GABAergic, and the cerebellar granule cells are the glutamatergic neurons. And you can see that astrocytes have a much bigger capacity for metabolizing all of these compounds than the neurons, again indicating that astrocytes are very important for brain metabolism. There is a very large capacity for metabolizing glutamate and glutamine, and also some of the tricarboxylic acid constituents such as malate. And another interesting observation that we did was that when we looked at glutamate and glutamine metabolism in astrocytes, we discovered that it is compartmentalized. Internally produced glutamine is metabolized differently from externally supplied glutamine. And I will go through this relatively rapidly because this is perhaps not that important, but the important thing is the next one. This slide is important because it shows the specific radioactivity of glutamate. This is the filled squares and glutamine. And as you all know, if you have a precursor product relationship in a metabolizing tissue, you can never have a higher specific activity in the product compared to the compound from which the product is produced. But nevertheless, the activity of glutamate was higher than that in glutamine, and this is something that is not possible. But that's what we found. So it is possible. And so the only way you can explain this is to imagine a complex model of glutamine and glutamate metabolism so that you have glutamate coming in to the cells, and it is in equilibrium with a metabolic a mitochondrial glutamate pool, and it is also in equilibrium with a glutamine pool that is localized in the cytoplasm. So by comparing all of this, we could come up with this model that clearly shows that glutamate and glutamine are not metabolized in a simple fashion in the astrocytes. And one would think that an astrocyte is a relatively simple cell, but it's not. It's a very complex cell. So that is probably why it's this way. Let's see. Yeah. This is, I think, one of the most important studies that I ever made together with Niels Diemer, Helene Benveniste. She was a medical student at the time. And we were the first to show that during ischemia, there is a very large increase in the extracellular concentration of glutamate. This is the content of a number of amino acids in the dialysate. You have this plastic membrane inserted through the brain. And you see that there is a lot of glutamine present in the brain, not surprising. There is also a very large concentration of taurine. And when we now looked at the effects of ischemia, I have to do it this way. This is the release of, come on. This is the release of glutamate and aspartate and glutamine. No, it's not. I have to go forward. Here we go. This is the release after 5 minutes or 10 minutes of ischemia. And you see that there is a very large increase in the extracellular concentration of glutamate and aspartate, and also of that of taurine. And we got this published in Journal Neurochemistry. They actually didn't like the paper, but they published it anyway. That's good for them because it has over 2,000 citations. You never know. So At the time, we did not understand taurine because some people thought that it was a neurotransmitter, which it is not. But the release of taurine has to do with the fact that when you insert or when you instate ischemia, there is a swelling of cells. And when cells swell, they release they release taurine. So, and the word microdialysis was actually coined in Copenhagen. It has never been used before, but it was used, I mean, it's used routinely. We should have patented the word. I would have been rich by now if I had done that. But I did not. So, and I'm not rich. So now I'm going into some slightly more modernized slides. So now we are out of the history. So now we are at present time. So I have presented to you already the glutamate uptake into astrocytes, and quite clearly, glutamate, which is released from glutamatergic neurons, acts on receptors, and it is, for all practical purposes, transferred primarily, not exclusively, but primarily into surrounding astrocytes. Glutamate expressing the GLT1 and the glassed versions of the glutamate transporters. Let's see if we can get to the next one, perhaps. Yeah. This brings me back to the anaplerosis. Anaplerosis means just filling up of things, and the major enzyme responsible for that in the brain is pyruvate carboxylase. So, since neurons do not express this enzyme, they cannot perform a net synthesis of glutamate because it requires that you replace alpha-ketoglutarate from the mitochondrial pool, and if you don't have any anaplerosis, that's not possible. So let's see if we can get the next one. Here we go. Jump to this one. One more. Here we go. So there has to be a complicated system that allows glutamine produced in astrocytes after glutamate is being taken up. The major part of glutamate is converted into glutamine, and that can be transferred by sodium-dependent neutral amino acid transporters, released from the astrocytes and taken up into surrounding glutamatergic neurons. And so this is the glutamate-glutamine cycle, which was coined many, many years before this. And interestingly, from the very beginning, it was thought that the glutamate-glutamine cycle functions in a stoichiometric fashion, so that all glutamate being released is taken up into astrocytes, converted into glutamine, and transferred back. So in a one-to-one relationship. That turns out not to be the case. There is some metabolism of glutamate to carbon dioxide in the astrocytes. So there has to be, even in the astrocytes, a way to compensate for this. And that is, again, dependent upon the expression of pyruvate carboxylase, which is exclusively expressed in the astrocytes, as I'm sure you recall. So now let's see. Yeah, I wanted to say a few words about ammonia, because in the process of the cycle, you have the glutamine synthetase and you have the phosphate-activated glutaminase in the neurons. So ammonia is used in the astrocytes and it is released in the phosphate-activated glutaminase reaction in the neurons. So what's going to happen to the ammonia? It could be just flowing around, but ammonia is not a nice substance. So there's probably mechanisms that are responsible for transferring ammonia from neurons to astrocytes. And there are several ways this can happen. One way is to utilize a keto acid. See. I hate this system. Okay. So one possibility is that you have a keto acid and an amino acid that sort of can be fluxed between one cell type to another. And one of the problems here is that glutamate dehydrogenase, thermodynamically, it is favoring glutamate synthesis. But in the brain, that does not happen because of the high Km value for ammonia and the high NAD/NADH ratio. So glutamate dehydrogenase works in direction of oxidizing glutamate to alpha-ketoglutarate and ammonia. So this is the complicated way that ammonia, or one way that ammonia can be transferred. Another way that this can happen is using lactate. Lactate can be exchanged over the cell membranes in exchange for pyruvate or together with pyruvate, and you have the pyruvate alanine aminotransferase responsible for this. And again, this will allow ammonia to be carried from the neuron to the astrocytes in the form of alanine. And that's much better than ammonia because alanine is not going to hurt anybody, and not the cells either. So that's another possibility. Next. Come on. So these are now the options. Come on. These are now the options. It can be the alanine shuttle, which I think has some importance. There can be a branched chain amino acid shuttle, which is principally similar to the alanine aspartate shuttle. And then there is aspartate, which may or may not be working. It probably is not. And then there is passive diffusion. That probably doesn't work either. So at the present time, nobody really knows exactly how ammonia is taken care of in the central nervous system. And if there is too much ammonia floating around, there will be serious consequences for the function of the brain, such as you see in hepatic encephalopathy. So that was a few words about ammonia. And then a little more metabolism. Again, pyruvate carboxylation and pyruvate and GD8. These are the two enzymes that can function as anaplerotic enzymes. Pyruvate carboxylation is not the only one. Glutamate dehydrogenase can also act as a way to increase the amount of tricarboxylic acid cycle constituents, because glutamate is now converted to alpha-ketoglutarate, and that will be taken up into the mitochondria and can function in the same way as pyruvate, which is carboxylated via pyruvate carboxylation. Yeah, let's try to look at this in a slightly different way. Yeah, I just wanted to show you how we actually do this, because you need a way to monitor transfer of carbon-13. We have in recent years we have always used carbon-13 instead of carbon-14, because you can if you use carbon-13 in connection with or in conjunction with NMR spectroscopy, you can actually see which carbon atoms are labeled. You cannot do that with carbon-14. And this method the NMR is not very sensitive. So we have combined this with mass spectrometry, which does not allow you to determine which carbon atom is labeled, but you can see how many carbon atoms are labeled. And if you look, for example, at the metabolism of uniformly labeled glutamate, you can see that you get this kind of labeling pattern in the tricarboxylic acid cycle constituents. And when glucose now is also used, non-labeled glucose, you get this complicated labeling pattern, and that can be utilized. You have 4 carbon atoms, 2 carbon atoms, 6 carbon atoms in the citrate, and that can be looked at using mass spectrometry. So it's a very powerful method to look at metabolism or metabolic functions in detail. Here we go. So glutamate metabolism, again, there's different possibilities. You can have complete oxidation of glutamate if you combine glutamic acid dehydrogenase with the conversion of malate to pyruvate, and then pyruvate can go back into the cycle as acetyl-CoA and can be metabolized to carbon dioxide. So this is the way that glutamate can be completely metabolized in the astrocytic tricarboxylic acid cycle. And actually, this is also how how we can get an increase in the amount of tricarboxylic acid constituents, either by pyruvate recycling that has to do with lactate production. So glutamate again is metabolized to pyruvate here, and it goes back into the cycle and will be metabolized to carbon dioxide. And then this is now combined with glutamate being converted into glutamine using ammonia. So this is another cartoon showing more or less the same thing, but it is not complete oxidation now of glutamate. Thanks. And it just shows that if you block the expression of glutamate dehydrogenase using siRNA technology, you can actually prevent glutamate from being metabolized, showing that glutamate dehydrogenase is an important enzyme for glutamate metabolism in the cells, something which sometimes have been controversial. Did I get the name? Yeah. So this is going to be, I think, my last slide. It's a little bit complicated, but it shows 3 different scenarios. One here shows complete oxidation of glutamate that I have already talked about. The next one is the so-called truncated tricarboxylic acid cycle, where glutamate is metabolized to oxaloacetate, and it comes back and will allow alanine to be produced. So that can be now in connection with the neurons. And then there is a third possibility, namely that the metabolism of glutamate via glutamate dehydrogenase can be coupled in such a way that you get an increased capacity for acetyl-CoA metabolism. So these are the 3 different ways that astrocytic tricarboxylic acid can function in collaboration with neurons, allowing exchange of glutamate and glutamine between the cells. And depending on which way this functions, you can have the possibility of utilizing glutamate in different ways. So it just shows how flexible the astrocytic metabolism is. And I think with this, probably is my last slide. I guess so. Yeah, let me again just point out that the collaborative studies that I have been doing over many years together with Leif Hertz and Ellen Kvamme and Gert Svenneby have been instrumental for my metabolic studies. And also, I want to mention that we have done this together with Ursula Sonnevald, Helle Våge Petersen, and Lasse Bach, Niels Vestergaard. Ursula is now retired. She actually lives in Copenhagen. Helle Våge Petersen has assumed a position as pro-rector for the university in Odense. And I don't know what that is. Presently, there's only a few people left in Copenhagen except myself, namely Anna Walls, Blanca Aldana, who actually originally came from Mexico, and then Jens Andersen. And this is the funding that I have been privileged enough to have been awarded for many, many years. The most important is the Lundbeck Foundation and the Danish Medical Research Council. So with this, I want to disappear. I want to thank you very much for your attention.
Well, thank you very much for this inspiring talk. We have some time for questions, if there is any. We can handle the microphones.
Don't be shy, because it's perfectly okay if you do not understand it, because I don't understand it either.
So is there any question? You know, I was wondering, Arne, that at the time that you started working, and I started as well, most of the work was done in male rats, right? I don't know if you did it in both males and females. So you did both. So do you have?
When you do tissue culture work, you have to use neonatal tissue.
Neonatal. Okay.
You can't do it. No.
Okay.
So it's either embryos or newborn. Astrocytes are from newborn brain, and the neurons are from embryos.
Right. So you don't know. So in theory.
So people would say, those that criticize tissue culture will say, well, these are all immature cells, so they cannot be used for anything.
Right.
But I'm not so sure that that's the case.
Okay. So any question? Yes, there's a question there. ¿Le pasas? Sí.
Hello, thank you for your talk. I was wondering what's your opinion about glutamate spillover? So you very well show how much of the glutamate goes to the astrocytes. So what do you think if there's a room for spillover of the glutamate?
I don't think the brain can tolerate a spillover of glutamate for reasons that I think I mentioned, namely that glutamate is a very powerful neurotoxin. So you cannot have glutamate floating around extracellularly, then the neurons will die. So there has to be means to get to metabolize, to take up glutamate primarily into the astrocytes, and glutamate can be metabolized, as I said, either to glutamine or to carbon dioxide. These are the options. And there is no other way that this can function, I don't think.
Any other question? Yes, there's one here. You can do it like.
I need to have you. I can't hear you.
If you could comment on the differences in kinetics between glutamine transporters into neurons terminals and glutamate transported from astrocytes?
Glutamine transport is not of high affinity. Glutamate transporters are high affinity transporters. So that, I think, is the major difference between the two. And the glutamine transporters are, they can work both ways. So that you can have release of glutamine or you can have uptake of glutamine, depending on the concentration. Glutamate can, under normal conditions, only be taken up into the cells, unless there is a metabolic deficit, and then the transporters that are energy requiring, they cannot function. So that's why ischemia leads to release of glutamate from astrocytes, or from neurons for that matter. And also please remember that transmitter release is energy dependent. You cannot have a release of neurotransmitter if there is no energy.
Any other question? Okay, so thank you again very much, ARNE, for coming down to Argentina. And now we hope that we can come some other day. Thank you. So now we go to the Young Investigator Talks. So who is the chair for this session?
Hello. So I didn't have enough time to see the TVs of the talkers, so we're going to change a little bit things. This is going to be like a contest. So I'm going to ask him who he is. So Young Investigator Talk number 1 will be Andrés Barani. What did you study, Andrés?
So in Argentina, I did my PhD, and then I moved to Paris to perform my postdoc. And finally, I got a position here in Argentina. And I started in 2020.
In which lab?
In Neural Lab and Belforte Lab.
Okay, so big clap for Andrés. And welcome.
Okay. Hola, hola. ¿Se escucha? No. So it's not working. Sorry. Yeah. Okay. The work I will present today, it was my project during my postdoc in Paris. So first of all, I would like to define what is motor skill. Motor skill is an action or sequence of actions which accuracy and or speed is improved by practice. So we have 3 main brain structures for learning: the cerebellum, basal ganglia, and cerebral cortex. So we have a dominant learning mechanism. In the case of cerebellum, we have a supervised learning where we have a nerve signal that we do something or change something within the cerebellum in order to generate an output. So then in the case of basal ganglia, what we have is a reinforcement learning where we have a reward signal that we do something within the basal ganglia. And finally, for cerebral cortex, what we have is an unsupervised learning. So these structures are involved in different motor functions. In the case of the cerebellum, we know that it's involved in online movement correction, motor adaptation, and short-term state prediction. While in the case of basal ganglia, what we know is that it's involved in motor vigor, motor program selection, and sequence learning. And finally, for cerebral cortex, what we know is that it's involved in state representation and predictive coding. So the 3 main players of motor learning are interconnected. In the past, it was believed that the cerebellum has reciprocal connections with the motor cortex via the motor thalamus. However, with the years, we realized that this circuit is more complex than that. In fact, the basal ganglia also form reciprocal loops with the motor cortex via the motor thalamus, as you can see. Oh, sorry. Oops. Okay. Finally, it has been recently established that the cerebellum and basal ganglia are reciprocally connected. So based on these studies, we can distinguish two different pathways, one which is composed by the cerebellum and the striatum, and the other pathway is the cerebellocortical pathway. So meta-analysis of motor learnings, including visual motor tasks, sequence learning, among other tasks, have shown a contrast of activation during tasks, where we can distinguish late versus early phases. In these pictures, we can see the three structures, cortex, basal ganglia, and cerebellum, and we can see how along the training, the activation is changing in both structures. In the three structures, sorry. So in summary, the involvement of the cortex, basal ganglia, and cerebellum evolves in the course of learning. The question is, do we have some interdependencies in these changes? So another thing that I would like to share with you is that the early but not late stimulation of the motor cortex erases a motor memory. So by using a repeated transcranial magnetic stimulation and a motor skill task, patients receive the stimulation right after the learning, and they observe in this article that the patients in the phase number 2, they were not able to remember what they had to do. So, and this was observed also for the phase number 3. However, if they let the patients to rest during 6 hours, and then they apply the stimulation, they can remember, suggesting that some of the changes in the role of brain regions occurs offline. So another thing that we know from the past is that in simple cases, such as sequential taping or visual motor adaptations, we have a separability of the involvement of these structures. For example, it is well known that for sequential taping, the striatum is the responsible. However, for the cerebellum, it is well known that the visual motor adaptation is dependent of this structure. So, but now we know that these two structures, basal ganglia and cerebellum, they interact each other. So our hypothesis is that the interdependency of the structures increases in complex tasks. So in summary, we know that motor skill learning involves multiple interconnected structures. Their involvement changes in time, and some of the changes occurs offline. And the degree of the interdependency of the structures may depend on the nature of the task. So based on these evidences, we decided to investigate or to see if we can reveal the interactions between the cerebellum and other foreign brain motor structures in the course of motor learning. So for that, we decided to use the rotor road in an accelerating mode. And here we can see the classical curve of learning, where we can distinguish the three different phases: acquisition, consolidation, and maintenance. Interestingly, this behavior depends on the cortex and basal ganglia, and more importantly for us, it was observed that the disruption of the cerebellar plasticity decreases learning. So in this study, we decided to focus on two pathways coupling the cerebellum to the cortex or the cerebellum to basal ganglia. And I would like to mention, that the cerebellum is composed by a cortex that sends, So the cerebellum sends inhibitory neurons to the deep cerebral nuclei, which are the outputs of the cerebellum. So here we have the deep cerebral nuclei. As I said before, these are the outputs of the cerebellum. and they send projections to the motor thalamus and the intralaminar thalamus. So we implanted animals and we recorded activity within the cerebellum, and we decided to focus on the dentate and interpose, two of the deep cerebellar nuclei. We have another one, which is the fastigial, but we didn't focus because this fastigial nucleus is not sending glutamatergic projections to the thalamic nuclei that we were interested on. So this is the protocol that I use. We have an open field session before and after the rotor road, and here in red we have the trials, and in blue we have the intertrials, which are the resting periods, and we repeated this protocol during 7 consecutive days. So this is the behavioral performance. We can see here the initial phase or acquisition phase, where we have a fast improvement of the behavior, followed by a refinement or consolidation phase, where the animals are still learning, but they remember what they have to do due to the savings. And then what we have is the maintenance phase, where the performance becomes asymptotic. So in terms of the modulation of firing rate of dentate and interposed neurons during the task, here we have examples of units in dentate on the left and interposed in the initial refinement and maintenance phase. And what we observe here is the activity during the open field sessions before and after, and the trials, and in black, the intertrials. And interestingly, we observe that only in the refinement phase and in the maintenance for both structures, we observe a decrease in the activity when the mouse is running on the road, okay? So the neuronal activity is selectively modulated during the accelerating rotor road task execution. So the next question was, okay, what about the distribution of the number of trials with significant modulations? For that, we compared the trial versus trial firing rate, and we observed a progressive increase in the number of trials, particularly in the refinement and maintenance phase. So then we wanted to see, or we were wondering about the cerebellar neurons display similar modulations during the accelerating rotor road task compared to open field locomotion. For that, we perform a linear regression where we plotted the firing rate versus the speed on the rotor road. And we observe that while the road was increasing in speed, so the fire rate decreased. And then when we analyze this modulation in the open field, we observe no modulation in the case of dentate, or maybe an increase, as you can see here, for the interposed nucleus. So then we plotted the same results for all neurons that were recorded in all animals, and we observed that every time in the initial refinement and maintenance phase, we have a down modulation for when the mouse is running on the road. And then, so the dentate and interposed nuclei are negatively correlated to speed, specifically on the road. So then, we wanted to see the effect of selecting those slopes that were significant, and we observed the pattern of activity across the protocol, across all the phases. And we observed that in the case of dentate, we found an increased down modulation during the maintenance phase compared to initial and refinement phases. And in the case of interpose, we observe stronger down modulations in the initial phase compared to the later phase, as you can see here, okay? In both cases, we still have the down modulation, down modulations produced by the rotor road, sorry. So then we wanted to make a perturbation in this circuit. For that, we decided to use a chemogenetic approach. We use inhibitory dread. This dread was injected within the cerebral nuclei. And in order to validate the protocol, we implanted animals and we recorded activity. We recorded activity in the 3 nuclei, and we observed that after CNO, by using 1 milligram per kilogram, in the 3 phases, we observed a decrease in the fire rate. So then we tested these animals in different behaviors in order to see if this perturbation could modify the motor function, since the cerebellum is highly implicated in motor activities. So we evaluated the force, the balance, and the ability to move through a vertical pole. Also, we quantify the steps, the foot, by using a footprint analysis. And finally, we measured the velocity in an open field, and we didn't observe significant differences between the experimental groups. So in terms of the motor skill learning, so if we inject these animals before the trial 1, at the beginning of the experiments, during these 7 consecutive days, we don't see significant differences in the initial phase between the experimental groups. However, we observe initial impairments followed by a relearning, as you can see here, in the consolidation phase or refinement phase. And in the case of maintenance phase, what we have is a global decrease of the performance. So then the question was, okay, this is an execution problem. So for that, I decided to use a fixed speed rotor rod by testing different speeds. And fortunately, we found that the performance was not affected. So then, as I said before, in the previous experiment, I injected CNO before the trial 1. So, but because CNO, the effect of CNO, it lasts maybe 2 or 3 hours, we didn't know if we were affecting also the consolidation phase, the consolidation of line. For that, we injected the CNO right after the trial 7, and we tested the animals. And what we observe is that we have an initial impairment followed by a relearning. So then we wanted to test a pathway-specific inhibition using threads again. For that, we injected a retrograde virus in the terminals. For that, it was the thalamus. And then we injected the dreads in the conditionally to express, to create expression, sorry, in the cerebral nuclei. And here we have, so here we have the pictures showing the injection sites, and we can see how the cells in the cerebral nuclei are labeled with the dreads. And we found if we inject in the case of the cerebral nuclei and intralaminar thalamus pathway, we found that if we inject the CNO before the trial 1, we have a global decrease of the performance, as you can see here. However, if we inject right after the trial 7, during the day number 2, 3, and 4, what we observe is no change. However, if we move to the other pathway, the central, the cerebral nuclei that goes to the motor thalamus, what we found is an initial impairment from day number 2 to day number 7, as you can see here, but every time the animals were able to learn. And finally, if we do the experiment where we inject CNO after the trial 7, what we have is again the same phenotype. So just to finish, the cerebellum interacts differently with the thalamus cortical and thalamus striatal pathway during the learning of a complex motor task, and perturbating offline the cerebellum-thalamic pathway prevents full consolidation of memory. Here we have a summary of the results, and you can see how the pathway that goes from the cerebellum to the motor thalamus is involved in the early phases of the consolidation phase. And finally, in the case of the cerebellar nuclei that goes to the intralaminar thalamus pathway, is involved in the online learning and also memory retrieval. The results about memory retrieval, I didn't have the chance to share with you. But if you have a question, don't hesitate to ask me. So just to finish, I would like to thank the directors of the lab, Daniela and Clement, and the collaborators, Roman, Caroline, and Jimena. So also I would like to thank to all of you and the organizers for giving me the chance to present my work. Thank you very much.
Hello, any questions? Thanks. No, we're not. No,
No questions?
maybe later.
It was clear, I guess. Yeah. So first of all, other animals, they are not able to perform this task. So it's difficult for them to run and to follow the speed of the road. So I was always using animals of 2 or 3 months old. You're welcome.
I have a question, but probably the answer is too long for what it remains. But so you show regions of the brain that if you impair their function, they affect the memory. But I guess behind that, there is the question of what is learned? Is the sensory or the motor output? So do they check how it works when they run? Or do they check some input from sensory feedback from what
I think it's a balance between two.
You think, well,
it's a balance between the two. So,
but is there any way you have a measure of how much the spinal cord affects? I mean, the actual, or
no,
do you have a way
that.
to mimic sensory input to? No. Okay, thank you.
We didn't check that. But it's a good question. Thank you. Okay, thank you very much.
Okay, and again, we're going to do the same now. Now it's contestant number 2, Antonella Rios.
Yes.
Hello.
Hi, hello everybody.
So what did you study?
I studied the role of one factor, which is ATP4 during DRG development.
So where do you come from?
I'm here from Argentina. I'm doing my postdoc at Fernanda's Leda's lab.
Okay, welcome.
Thank you. Yes. Well, hello everybody. Thanks to be here. I'm Antonella, and the title of my talk is ATP4 regulates peripheral innervation and the development of peptidergic sensory neurons mediating pain stimuli. So, sorry. Okay. As an introduction, DRG primary sensory neurons are pseudo-unipolar neurons that have one axon that duplicates, and one of these branches goes to the peripheral target, such as skin or muscles, and the other branch innervates the spinal cord. These DRG primary sensory neurons could be classified according of their perception modalities, and these correlate with the expression of specific neurotrophic factor receptors. For example, proprioceptors are neurons that respond to NT3 and express the receptor TrkC. Mechanoreceptors respond to BDNF and express the TrkB receptor. And most important for this talk are the nociceptors, which are neurons that mediate noxious stimuli. And this class of neurons could be classified into non-peptidergic neurons, which respond to GDNF and express the receptor RED. And these neurons mediate cold pain stimuli. And the nociceptors, which are peptidergic neurons that express the NGF receptor TrkA. These neurons innervate the epidermis, and I'm going to focus in this talk, in particular in the nociceptors that respond to NGF, innervate the epidermis, and mediate noxious heat stimuli. So during early development, NGF TrkA signal is necessary for peripheral innervation, axonal growth, for the survival of the cells, and also for the induction of different molecules that could be mediating all these biological effects. And several studies indicate that animals lacking NGF or TrkA have defects in peripheral innervation and also in the transduction of pain stimuli. So the aim of the work of our lab is to understand which are the molecules that are downstream NGF TrkA signaling that could be mediating all these biological effects. And previously work of our lab demonstrate that ETB4 and ETB5, which are two transcription factors that belongs to the PEA3 subfamily of ETS transcription factors, are induced by NGF in DRG sensory neurons. And these transcription factors have an ETS motif, which is the DNA binding domain, and are known to be oncoproteins because its expression is dysregulated in several tumors. Previously work of our lab demonstrate that ETB4 and ETB5 are expressed in DRG TrkA positive neurons during early development, and when we downregulate the expression of these transcription factors, DRG rat sensory neurons are shorter in culture. I'm going to focus in ETB4, and the aim of this talk is to understand if ETB4 has a physiological role in DRG sensory neurons. So, as I mentioned in the introduction, something is wrong with the bar graph. Presentation. No. Well, there's a problem with the bueno. No sé por qué no se ven los gráficos. No se ven los gráficos. No se ven los gráficos. Sorry about that. Los gráficos que sí se verían. Well, you must believe me, but sorry about that. As I told you at the introduction, nociceptor innervate the epidermis, and we analyzed what happened with peptidergic innervation in ETB4 control and ETB4 knockout mice. And we did it by immunofluorescence against CGRP, which is a peptide that is expressed in peptidergic neurons. And well, you cannot see here, but there's a decrease in the number of CGRP free nerve endings innervating the epidermis derived from ETB4 knockout mice. And when we measure the length of these neurites, there's a reduction in the length of these free nerve endings derived from ETB4 knockout mice. We wanted to know if the same occurs with non-peptidergic innervation. For this end, we analyzed and the GFR alpha 1 and GFR alpha 2 projections, which both are the co-receptor FRET that are expressed in non-peptidergic neurons. And we didn't see any differences between both genotypes. So ETB4 is required for the outgrowth of peripheral projections in DRG peptidergic neurons. All the present, yeah. So these problems in ETB4 in ETB4 deficient animals in peptidergic innervation could be due to a reduction in the number of neurons in DRG, and we analyzed that, and as you could see here, there is a reduction. There's no differences in the number of neurons in DRG ganglion between both genotypes, and there's no differences in the number of TRK-positive neurons. And we analyzed the same by the analysis of the mRNA expression levels of these receptors, and similar conclusions were obtained. So we analyzed the axonal growth of these neurons, and for this we performed different cultures from DRG and from the another sensory ganglion, which is the trigeminal ganglion. And there's when we measure the length of these neurids, as you can see here, there's a reduction in the length of the neurids from culture from ETB4 deficient mice. So these developing neurons had a dynamic structure at the tip of the axon that allowed the cells to innervate peripheral tissues, and this structure is the axonal growth cone. So we analyzed the structure in these cultures, and as you can see here, there's a reduction in the growth cone area in neurons derived from ETB4 mice. So ETB4 regulates axonal growth and axonal growth formation in sensory neurons. And the question is how this transcription factor mediates this effect. And there are several studies that indicate that PA3 transcription factor control the expression of a group of metalloproteinases, which I'm going to refer to MMPs for the rest of the talk. And these MMPs are necessary for extracellular matrix remodeling proteins that are necessary for the axonal growth and innervation of the tissues. And when we use an inhibitor, when we use an inhibitor of these MMPs, as you can see here, there's a reduction in the length of the neurids, and also the axonal growth cone of these cells is shorter compared to those that were not treated with the inhibitor. So, and this phenotype is similar that we observed in ETB4 deficient neurons. So we analyzed the expression of a group of MMPs in ETB4 knockout mice compared to control in DRGs. And as you can see here, NGF induced expression of a group of MMPs in the control situation, but not in those DRGs derived from ETB4 knockout mice. And this phenotype is reversed by exogenous supply of MMPs in culture, as you can see here. Well, you can pass on my poster later to understand all the paragraphs that aren't here, but as I mentioned in the introduction, nociceptors are those neurons that mediate pain stimuli, and in particular, noxious, those that express the trachea receptor, which are the peptidergic neurons, mediate noxious heat stimuli. So we want to understand if ETB4 mediates this transduction of pain, and to do that, we performed different tests, and what we saw was that ETB4 deficient animals are less sensitive to noxious heat stimuli at these temperatures, but not at 4 degrees, which is a temperature that is cold temperature that is sensed by non-peptidergic neurons. We observe similar results by other tests, which is the formalin injection test, which measure the response to a chemical study, and ETB4 knockout animals were less sensitive to formalin. And no differences between both genotypes were observed in the von Frey test, in which we measure mechanical stimuli. So ETB4 mediates is a mediator of the nociceptor responses induced by NGF-TRK in sensory neurons. So you probably know these two people who won the Nobel Prize last year because of the discovery of the temperature receptors. And these ion channels are expressed in peptidergic and non-peptidergic neurons. So we wanted to know if ETB4 is necessary for the expression of some of these channels. And to do that, we analyzed the mRNA expression of a group of ion channels, and we saw that ETB4 mediates the expression of TRPV1 and TRPV2, which are those both channels expressed in peptidergic nociceptor and mediate noxious heat stimuli. We go deeper in the analysis of TRPV1, and by immunofluorescence, there's a reduction in the number of TRPV1 positive neurons in DRG from ETB4 knockout mice, and this channel is sensitive to capsaicin, which is the compound of the chili pepper. And when we inject this compound into the hippo of ETB4 and control mice, there's a reduction. These animals, knockout animals, are less sensitive to capsaicin. So, in summary, during development in the NGF induced expression of ETB4 in DRG sensory neurons, and this transcription factor is necessary for axonal growth and for TRPV1 expression in that neurons. And this is because this transcription factor regulate the expression of different MMPs, which are necessary for peripheral innervation. When ETB4 is absent, there's a decrease in the peripheral innervation of proprioceptive peptidergic neurons, and a decrease in the expression of TRPV1. So animals lacking ETB4 are less sensitive to noxious heat stimuli. I would like to thank everybody to be here and to trust in what I said before. And I would like to thank my mentor, Fernanda Leda, and Gustavo Paracha, and all the members of the group. Of course, I would like to thank our collaborators, Pablo Brumowski and Jorge Aquino, and all the organizers of the meeting. And you can pass from to my poster tomorrow, please, so to see all the graphs that were not here. Thank you.
Hello, any questions? Allá.
Hi, Cristian
Hi.
Gonzalez, University of Chile. Very nice work in spite of the technical things, right?
Thank you.
So I have a couple of questions. One is kind of a technical question, and it is when you address the effect of MMPs in your in vitro system, what are the components of the extracellular matrix that should be degraded by the MMPs?
Cultures were done in a polyornithine cellular matrix and laminin, and probably this is sufficient to show all the effect that we saw in the axonal growth one when we add not only the MMPs inhibitor GM, but also in knockout mice.
And the other is related with the role of TRPV1, because you showed that staining of cell bodies on DRGs. Do you think that TRPV are also expressed in the axons, and that have a local role, and in that case, the calcium influx by TRPV may be modulating, for example, actin or microtubule dynamics?
Yes, TRP1 is expressed in the axon of these neurons. We didn't evaluate that because of technical problems with the staining, but I expect that there was a reduction in TRPV1 nerves in the epidermis, and that could be mediating all the effects that we observe in the behavioral.
Hi, here.
Oh.
Very nice talk, thank you. I
Thank
was
you.
just curious, like you mentioned the sensitivity to capsaicin at the end, and you said you measured after injection. I was just curious, how did you do that?
Yes, this work was done in collaboration with Pablo Brumowski lab, and what they did was inject capsaicin into the himpo of the legs of these animals and evaluate the time that they pass doing nociceptive response in a period of time. So what we evaluate is the time that the animal is sensing the pain to this compound, and that's what I quantify in the graph.
Thank you.
Hello, anyone? No, Diego.
Okay, so now I have so treatment the same pathway fluorination.
Yes, I think that TTP4 could be mediating the origination of the axons after a crash injury, for example. We have some preliminary experiment that could indicate that because when we make a sciatic nerve injury, ETP4 is induced in some preliminary experiment in DRG cells. So that could be a reason that ETP4 could be playing a role in regeneration of the axon. Thank you.
Hi. I was wondering, have you noticed any changes in the size of the nociceptive field area when you tested the knockout mice and the other ones?
I didn't check that. I could check that from the images, but no, I didn't see anything wrong or that I make me curious about that. I count the number of these neurons, but not the size of if they're bigger or shorter.
Oh, okay. Yes. No.
Okay, thank you very much.
So thank you for your attention. Now we have more or less an hour, more than an hour probably for lunch. We have to be here back in at 1:30. Yes, 1:30? No, 2:30. Sorry. And what I was just been told is that for next speakers, if they can please bring their presentation beforehand so that these kind of technical problems that we apologize can be solved beforehand. And if you can have it both in PowerPoint and in PDF, it might be better. So